Hi, everyone. Good morning or good evening, and it's almost good night from Japan. So I'm Kazuyama from Osaka University, Japan, and I'm so excited to host today's speaker, Gerard Eberl, because I'm big fan of his study because I also work on ILC2, but he have done so many beautiful work on ILC3. So I'm very honored to host his talk. I used to host many of speaker from Asia because of the time difference, but I didn't sleep today and wait for this timing because I really wanted to host his talk. So I'm very honored to introduce Jalal before his talk. So Jalal completed his PhD at the University of Lausanne on the structure of epitope recognized by T cell, followed by a first postdoctoral fellowship at the Ludwig Institute for Cancer Research, also in Lausanne, and on the development and function of invariant NKT cells. Second postdoctorate in New York led him to characterize the role of nuclear hormone receptor, rho gamma T, in ILCs. I think this finding lead him to the field of ILC3 for so many years. And in 2005, he was recruited to the Institute Pasteur in Paris to lead the lymphoid tissue development unit, which became the microenvironment and immunity unit in 2015. And for 10 years now, his laboratory has been studying the impact of symbiotic microbiota on the development and activity of the immune system, mainly in the gut, but also in the adipose tissue and also sometimes in the lung. And more recently, he has been interested in the understanding how the microbiota also influence mouse behavior. This is quite interesting. Through the activation of innate immune system and how the nerve system modulate immune responses. So I think he gonna talk about this, hopefully. And from 2015 to 19, Jalal was a director of the immunology department at Institute Pasteur. So Jalal, I'm so happy to have you in this global immune talk. And before having your talk, we always want to know your personality a little bit more before your talk. So I want to ask one question before your talk. And today's question is, what is a trait in your personality that helped you the most in your scientific career is what I want to ask you first.
So, Kazuo, thank you very much for the invitation, and thank you very much for this beautiful introduction, and thank you all for being here today. So yeah, thank you for the question. It's an interesting question because I guess you cannot really answer the question without a little bit of narcissism, since the question is about, what are the traits you think are important in yourself. But so I don't have an answer about me, but I have an answer about what I think are important traits in not only in scientists but in general to, I guess, to achieve some level of success. And I hope that I have a little bit of these qualities in me. So I guess one is, and I guess you'll agree, is resilience and perseverance. And especially in science, we have to stomach so many failures. Most of the experiments fail. That's totally normal. And just to stomach them and to be ready for when it works, to be really ready for it works and recognize the success is very important. And again, it's not something which is only for science. The other thing I think which is key is curiosity, and also that's obvious in every branch of life, you know, asking questions and why the hell is it this way, that way, helps you to be creative and go forward and create new things. And the other thing which I think is key is ego. You know, I guess we see ego as probably a negative thing, but in its limits, ego is absolutely necessary, which, you know, it means that ego means that you have to recognize the qualities in you which make you special, and we're all different. We have all different qualities, and it's not always easy to recognize our own qualities because you're in societies with different norms, and you're judged by these norms, and sometimes you're not normative and you judge negatively. But you know, it's important that every one of us believes in their own qualities and nurture them to be successful in whatever we do. So I hope that I have a little bit of these, which helps me to go forward.
Okay, thank you very much. Okay, so let's move on your talk. And I heard your title is Regulation of Hypothalamic Function by Microbiota. That's correct? Okay, so if you are ready, could you please share your slide?
All right, so here we go. And do you see the slide?
Yes, cool. It's okay.
And you see the
yeah.
arrow?
It's not a pointer, but it's an arrowhead. Yeah.
Should I change the point? I'm not sure. Maybe I should.
Maybe it helped listener.
Okay.
Yes.
Okay.
Yeah, both are okay. Yeah. Thank you very much.
Thank you, Kazuo. Very kind. Thank you all for being here, and it's really a pleasure and honor to speak in the series. And I saw in the list of attendees many of my friends, and I would love to be, you know, with you speaking about stuff. But it's still a pleasure today to talk about what we do in the lab. So I'm going to focus, as Kazuo said, more about recent work we do together with our neuroscientist colleagues, Pasteur, and that is the impact of the microbiota and immunity on the hypothalamus. So it's really a venture between three labs. Since I arrived at Pasteur, I'm interacting a lot with microbiologists. So we are the immunologists, obviously, in this trio, and we work since ever with the lab of Ivo Bonecca, who is a biochemist of the bacterial cell wall. And now, since 6, 7, 8 years, we work kind of in synergy with the lab of Pierre Magliedo and Gabriel Lepousset, who are genuine neuroscientists, neurophysiologists, and it has been so far an incredible adventure to discover, to explore what the brain does together with the immune system. So as an introduction, just let me tell you a few things about how I look at the microbiota, and just to tell you a few things about what I'm not going to talk to you about today, but is an important part of my lab, and then focus on the impact of the microbiota on hypothalamus. So I'd like to see the microbiota colonizing us at birth, not necessarily as, you know, as immunologists like to say, you know, pathobionts or potential dangerous entities, but more like cells which bring new functions to the some people call that holobiont or superorganism. And obviously since 10-15 years it has been an incredible field to understand what these functions are for the host. And of course, like our own cells, these cells can be dangerous. Our own cells can make cancer. These cells can infect us and expand as well. But here it's really about the functions they bring to the host. And an area which we have been exploring, and in which many people are now involved, is the impact of the microbiota before birth or just after birth and before doing weaning on the ontogeny or the programming of the immune system. So before birth, a few labs have looked at the impact of the metabolites produced by the microbiota of the mother. And Andrew McPherson's lab in Bern has shown a number of things in that area. Yasmin Belkaid's lab has shown impact of infections of the mother on the programming of the ontogeny of the immune system, or let's say imprinting. I will come back to the word imprinting. Many labs have shown how the colonizing microbiota after birth impacts on the ontogeny or the regulation of the immune system in the long term, or even the microbiota itself, depending on the context, can become kind of dysbiotic or different in a stable way, and thereby impact then the susceptibility to disease. We have been more active in doing the weaning to show that bacterial products will impact on the reactivity of the immune system in the long term, and therefore determine the susceptibility of the host to inflammatory disease. What I've shown with Ziad Al-Abani, who is here today, we have shown that the weaning is an important period in that process, because when the mouse, or well, the mammals in general, go from a milk-only diet to a diversified diet, a number of bacteria can now expand thanks to new nutrients, and we see that there's something like 100-fold increase in the number of bacteria in the gut, and that is met obviously by a strong immune response. But what Ziad saw is that there's a time frame during which this response will have an impact long-term on the immune system. It cannot occur before, because as Catherine Knoop showed, and she's also in the audience, showed in her studies, is that before this impact of the microbiota on the immune system is blocked by things like EGF in the milk, which blocks the transfer of antigens to goblet cells from the lumen into the parenchyma of the gut. And it's not totally clear, even though Catherine suggested that it's still are mediated. After weaning, also there is kind of a closure of what we like to call the window of opportunity. Nonetheless, so what we showed is that this weaning reaction is going to imprint the reactivity of the immune system in the long term. That is, if the weaning reaction is strong, the reactivity of the immune system in the long term is going to be pretty low. And the converse is that when you block or you kill the bacteria with antibiotics, or you block the weaning reaction, so a low response is going to be induce a high reactivity of the immune system in the long term, and therefore what Ziad showed, high susceptibility to pathologic inflammation. So in this graphical abstract, I've probably pushed a little bit the arguments, because it's not necessarily pathologic, it's just increased susceptibility, which might in some contexts being beneficial, for example, when exposed to infection. But that's still something we are defining. So why we call that imprinting? It's going back to the notion of imprinting as defined by Conrad Lorenz in geese. And what he showed is that the first animal sound these geese hear is going to be associated, when they hatch, is going to be associated for life to the mother. So this lady here has probably talked to these hatching geese, and therefore she becomes their mother for life, and that has allowed people to study geese, for example, migration. And it's quite a phenomenon. So it's motherly imprinting, which we also witness in ourselves. For accents, it's very difficult to get rid of accents from one language to the other, because apparently the baby has to hear sounds and train the sounds before year one to be able to spell them out correctly. So that's also a form of imprinting. Of course, here we talk about immunological imprinting. So this is half of the lab works on the how this neonatal immunological imprinting is induced by bacteria, what's the metabolites which are involved, how does that work, which are the cells involved, probably any type of cell, at least in the gut and beyond, is involved at some level in this neonatal imprinting. We're looking at, as many have done before, at epigenetic imprinting and the consequences of it as I said before, on susceptibility to disease or resistance to infection. So going back to a work we did very early on when I arrived at Pasteur with my colleague in microbiology, we looked at the second week, the first 2 weeks of colonization of the mouse gut by microbiota, and we compared for reasons of what people had shown before in the field, that we looked at the development of what was called isolated lymphoid follicles. So you can see here a mature ILF, which basically includes a rim or a capsule of dendritic cells. In green, you have lymphoid tissue inducer cells, a subset of ILC3s, and in red, B cells. So in germ-free mouse, this does not, these ILFs do not develop. What you have are just cryptopatches. So cryptopatches have been thought before to be the site of extrathalamic generation of T cells. We showed when I was in Dan Littman's lab that these cryptopatches were, these green cells were ILC3s, lymphoid tissue inducer cells, basically the same as those which induce development of lymph nodes and pass patches in the embryo. The difference is that lymph node and pass patches are programmed to develop while the ILFs are not. So in germ-free, these do not develop and cryptopatches stay put, and bacteria somehow induce the CPs to collect B cells and make a mature ILF. So what we showed with our microbiologist colleagues is that epithelial cells using this innate receptor node 1 detect product of proliferation of bacteria, which are muropeptides. I will come to that. But so these muropeptides are an easy or raw readout of the activity of bacteria in terms of proliferation. And so when these epithelial cells get activated, they produce chemokines and this defensin, which actually can act as a chemokine and bind CCR6. So by binding CCR6, they will activate LTI cells, which in turn activate stroma cells, which in turn collect B cells, which in turn will do IgA against the bacteria, and therefore establishing a negative feedback loop on the inducer of the process. So the more bacteria, the more induction of ILFs, the more ILFs, the more IgA, the less bacteria. And there is an equilibrium which is reached at some point. And if you block this process making ILFs, then the result is a 100-fold increase in bacteria. So why do I go back to this now old stories? Because, well, you will see why. So coming back to the bacteria, you know that bacteria have a bacterial cell, have a cell wall, which consists basically of a huge molecule of sugar, different sugar sheets which are bound between them by these little peptides. So this peptide glycan has to be deconstructed when bacteria proliferate, and one way to do it is to cut the sugars, the sugar moieties, and the bacteria will release these small peptides, which are D3 or tetrapeptides, bound to 1 or 2 sugar moieties in the environment, and they can reuse them after division to rebuild their wall, but a fair amount of these gets lost into the environment, and that's exactly what a node 1, node 2, and other receptors detect. And by the way, this detection of these muropeptides is quite universal. It's been first described in plants, And then by sequence homology found in mammals, it's also found with different receptors in Drosophila and other types of metazoans. So in mammals, the receptors, two of the receptors are node 1 and node 2, which are intracellular. So they will detect proliferating bacteria in the cytoplasm. They will also detect muropeptides, which are pumped into the cell by some transporters. And so once node 1 or node 2 activated, they will induce the NF-kappa B pathway, but also autophagy, which will help to get rid of the bacteria. So it's a small molecule, D2 tetrapeptide. It's very soluble, so it's going to reach the bloodstream fairly easily. And once in the bloodstream, can go anywhere. It has been shown 10 years ago that muropeptides have an impact on the generation of granulocytes from the bone marrow and increase the resistance to bacterial infection. It has also been shown to be involved in the maturation of the brain. So one thing we investigated was whether muropeptides can transport information from the gut to the brain in the same spirit. That is, it carries information about the proliferative activity of the bacteria, and the question here was, is that going to be read out as well by the brain? So it's not a question which came out, in the shower or in the bed at night. It's a question which came out from all the papers in the 70s. This group has shown that, or has looked for factors in the blood which were correlated with sleeping disorders, and they isolated a factor called factor S for sleeping disorder, and looking at the structure of it, found that it was a muropeptide peptide, which is one type of muropeptide. And when they injected this NDP into the brain of rats, these rats did have a modified sleeping pattern. So we started the whole project this way. We didn't inject in the brain, we injected IP, these muropeptides, and indeed we saw differences in sleeping patterns, which I don't show here, which we did not publish because it worked for 1 year or two, but then was very difficult to reproduce because you probably know that the sleeping pattern of mice is quite chaotic. Hours, I think human is cycles of 1 or 2 hours. In mouse, it's much shorter. In something like 10-15 minutes of deep sleep, REM sleep, and wake, and it becomes very chaotic and dependent on the condition, the animal facility. And then we shifted to metabolism, as I will talk to you about. So first experiment we did was to really test whether muropeptides produced in the gut could actually reach the brain. And so what our colleagues in microbiology did was to incorporate radioactivity into the bacterial cell wall of E. coli, then colonize germ-free mouse with that, and after 24 hours, take organs of these mice and check radioactivity. And we could see indeed, well, as expected, we could see radioactivity and presumably muropeptides in the blood, but also fair amounts in the brain. But really, the result which kicked off the project was looking at the brain. And so what you see is a coronal section of the brain. So you cut the brain this way. And this is a brain of a NOT2 GFP reporter mouse. So NOT2 is expressed widely in macrophages, in microglia, in the gut. It's primarily macrophages. It's full of it, of course, because they're going to read out directly in situ the proliferation of bacteria. So for those not familiar with the brain, so this is the cortex. This is the hippocampus, the thalamus, the hypothalamus, and the striatum. So you probably know that the hippocampus is where you form short-term memory, cortex, long-term memory. The thalamus is kind of guard, the triage for sensory inputs, and the hypothalamus, which I'm going to focus about, is more about physiological regulation, food intake, sexual activity, and a number of other things, body temperature. So we were kind of entranced by this wide expression of GFP in the brain. You don't see so much in the hypothalamus, even though I'm going to focus on the hypothalamus. But it was already an incredible surprise to see that. But then I became skeptical, and probably it's going to be microglia as expected. And so when Ilana Gabani, who did the work, looked closer, she saw a fair amount of expression in the hypothalamus, and in the hypothalamus, actually in every region, looking at which cells expressed GFP and NOTCH2, it was again expected that microglia, here stained by ABA1, would express GFP. So you can see those guys expressing GFP here on the right in the overlay. But it was much more a surprise to see actually neurons express GFP quite widely in the striatum and the thalamus, and to some extent also in the hypothalamus. So that was a surprise, and of course we want to be sure that this serves a purpose or has an impact. And Ilana crossed NOTCH2 flox mice to VGAT3 and CAMK2 mice. So VGAT is expressed in activating neurons, and CAMK2, sorry, it's the opposite. VGAT3 is expressed in inhibitory neurons, and CAMK2 in activating neurons. And I will talk only about the VGAT3 mice because that's where we found a phenotype in inhibitory neurons. So this is the weight of mice with age. This is a wild-type mouse, so you can see females take some weight, then they control their weights until some point, while the males, as we know, take on fat and fats without any control. Now, in the conditional knockouts of mice which do not express NOT2 in these inhibitory neurons, all neurons, as expected, no phenotype in the males because there is no control in the weight increase. There is also loss of control in weight increase in females, which is due to increased feeding in this knockout. And you can do actually the opposite experiments where now you feed NDP, so that's gavage. You gavage NDP, so that's a muropeptide, and you inhibit the food intake. So it goes both ways. The absence of it increases the food intake, or gavage with NDP decreases the food intake. So this is about taking in energy. What about expenditure of energy? And it's actually the same. So in control mouse, if you give them a coton in the cage, they will make a nest so that they can sleep together and save energy during the rest period. This mouse here without the NOTCH2 in these neurons doesn't care. It has eaten more, it has more energy, and who cares about spending more since you have more, basically. So that's you can see that difference here. And also in terms of not only behavior, but in terms of body temperature, a wild-type mouse is going to have to decrease the body temperature from activity to sleep by something like between 2.5 and 3 degrees. So that's nighttime temperature, 37 or something. That's actually here, 37. And then it decreases to 34 during daytime for us and rest time for them. And this difference is significantly less in mice which do not express NOT2 in these neurons. Now looking at the neurons' activity themselves in the brain, and the way to do that is to look at CFOS expression in neurons. So CFOS is an early activation marker in neurons. And what Ilana did was to compare wild-type mice to these conditional knockouts and to look which are the nuclei, that is the group of neurons in the brain which have differential activity And we were pleased to see that these three nuclei, that is the arcade nucleus, the lateral hypothalamus, and this one I can't remember what it is, but all these three regions are in the hypothalamus and are involved in food intake and body temperature control. That totally fitted the phenotype we found. So that's in the knockouts now, feeding again, doing the reverse experiment, feeding MDP to the mice and looking at the activity and do some imaging statistics. We could see the same, that is, that's the hypothalamus. So we are at the bottom of the brain. This is called the third ventricle. So the arcade nucleus here, the DMH, you can see a depression of activity. And this is an increase in activity in that region, which is, so you would probably ask me the question why, which is only found, well, I already told you that the phenotype is only in females, but furthermore, it's only found in all the females, that is females which are I can't remember the term well, which are not fertile anymore. Okay, so why don't we go and listen to these neurons? That's the reason why I start to love neuroscience, because you can do things which are really cool, listen to the activity of the cells, and you will see knockouts, regionally the activity of the cells, things which are arguably harder to do with the immune system. So to listen to neurons, what you do, you insert an optical fiber in the region you want to listen to, and then you have to have a reporter for activity. And so you are going to insert this receptor for calcium, which turns on fluorescence when bound to calcium. And how you're going to do that is infecting that region, and that region only, with an adeno-associated virus, an AAV, which expresses this thing, and you can make it very specific. This is going to be only expressed in neurons of interest, and because this is a conditional expressor, and infecting VGAT3 mice. So this report is only going to be expressed in these VGAT neurons in the hypothalamus. So when you listen to a mouse before feeding, that's the activity of that region. So that's a regional activity. What you see is a population activity of neurons. And after feeding, this activity is nullified, is totally inhibited. Okay, so far so good. Now we fed the mice with a control, that is a muropeptide, which looks like the ligand of NODS2, but is not a ligand. And so the experimental control is the procedure does not inhibit the activity of these neurons. Now feeding MDP does indeed inhibit the activity of these neurons. And now I'm not going to show you statistics, but of course we did. The other thing we did was, okay, so it looks like feeding the MDP to the neurons presumably makes MDP travel to these neurons, inhibit the activity directly, but we didn't really show that, and that's what is shown here. So what Ilana did with the colleagues in neuroscience was to do a patch clamp of neurons in thick sections of the hypothalamus. So this is a neuron where you're going to insert a pipette and measure the potential between the inside and the outside. And then you can also inject stuff in the neuron to see the effect. So the control MDP or the MDP, and measure the polarization of these neurons. So this is typically the much cleaner activation readout of a neuron. This is population, and this is a single neuron depolarization cycle. So what we showed here is that doing the procedure with the control MDP will not have an effect on the neurons. This decrease is probably because of the procedure itself, but injecting NDP directly in neurons do quite efficiently inhibit the activity of these neurons. So that was pretty cool. So the MDP directly does block the activity of these neurons. Now, what we wanted to finally do in this last experiment was to, remember we did a conditional knockout of NOT2 in these inhibitory neurons. Now we wanted to do the same, but just in the inhibitory neurons of the hypothalamus. Again, that's something which is pretty cool to do in neuroscience, probably impossible to do in immunology. And the way you do is pretty similar to what I told you before, is to inject an AAV expressing CRE under control of, it's not written here, but of a promoter which is only expressed in the neurons of interest, that is VGAT, in this conditional, in this flux NOT2 mice. So you will only knock out NOT2 in these neurons in that region. And when we did that and looked for the phenotype, we recapitulated exactly the same things as I showed you before. That is a loss of control in weight increase, in food intake, in control of body temperature. So that was nice. And in that last experiment, what Ilana did was to give antibiotics to the mice to show that it was driven by bacteria in the gut. And as you can see, giving antibiotics to the mice does nullify the phenotype of food intake control, and removing the antibiotics leads to a regain of control in food intake. So it's really the microbiota and gut, and all the experiments we did with Muropeptides, when we injected them, we didn't inject them, we garbaged the mice. So it really goes from the gut to the brain. So in summary, what we showed in this study was that when bacteria proliferate in the gut, they're going to inevitably release muropeptides. So these muropeptides are going to go in the circulation in the blood, and they will reach the hypothalamus, where they will inhibit the activity of inhibitory neurons controlling food intake and body temperature. So why does that happen? What's the purpose of it? So one hypothesis is that it's just another way to control food intake, like leptin, and we could not find an instance where it acts in the place of leptin, but it's possible that there are some contexts where leptin is not sufficient, not operative, and now this system can work. I would propose another hypothesis, is that linked to what the brain can do in sensing food. So I would suggest, and this is really just a hypothesis, that excessive bacterial proliferation due to food intake might be dangerous for homeostasis. So the hypothalamus might be sensitive to that and react to an increase in the MDP because of excessive proliferation, and associate that to food intake. So decrease food intake to avoid this dangerous situation, or more specifically, to avoid eating this type of food. The brain will have ways to judge, to classify the type of food the mouse or you eat, and associate that to increase the levels of NDP. And maybe that translates into not just satiety, but disgust from that type of food, because the brain associates that to a dangerous situation. So I like this hypothesis because it's more sexy. It goes back to a little bit of Pavlovian, a regulation of food intake, but that's something we have to test. So let me shift a little bit gears and talk to you about the HPA axis. And but again, it's about how the microbiota via the immune system will have an impact on the activity of the hypothalamus. So the HPA axis is the hypothalamus-pituitary-adrenal axis, which is kind of the stress axis. So when the brain senses stress, so by stress I mean whatever you mean, psychological stress, physical stress, immunological stress, this is going to feed the nuclei, a nucleus in the hypothalamus called the paraventricular nucleus, the PVN, to activate neurons which produce CRH. And CRH is going to go in the circulation portal to the pituitary glands, and pituitary gland cells are going to produce ACTH, which will, through the bloodstream, reach the adrenal glands, and there the cells will produce cortisol or corticosterone in mouse. So what Ferdinand did, a former PhD student in the lab, was to look at the circuitry which is induced in the brain by LPS. So LPS is ever present in the blood. You can measure LPS at a steady state in the blood. Of course, it's going to increase if there is a breach or a decrease in an increase in gut permeability or an infection. And this leads to lots of cytokine production, among them iron beta, TNF. And Ferdinand injected a lot of different cytokines, but really this axis of LPS, iron beta, TNF was the most powerful to induce the HPA axis. And what you see here is this Christmas tree is the activation map of the brain upon LPS administration to a mouse. And this is measured 3 hours after IP injection of LPS. So he measured 2 things: the cortisol production, that is activation of the HPA axis, and the activation by CFOS of neurons in the brain. So the size of the circle is proportional to the number of neurons which are expressing CFOS in these different nuclei. So you can see LPS and beta is basically quite similar in activation potential of these centers. So that's the mouse brain. This is the cortex here, the cerebellum. The hypothalamus is here. The acute nucleus we talked about is here. The PVN, which is not really here, is really here, is the master of the HPA axis. And this is basically the input region. For example, the vagus nerve is going to feed into the NTS. And there are other regions like here, where the brain parenchyma is, or let's say it's a very vascularized region where the brain via stroma cells or endothelial cells can directly sense information from the blood, and there's a system then to translate this information into activation of neurons locally. Anyway, so information floods from the periphery into this region, which then transits by different circuits into in different circuits and different impacts on the brain, and among them the activation of the PVN. So you can see that upon LPS, iron beta, TNF administration, the PVN is hugely activated, feeding into the HPA axis. The NTS is activated because that's the input. Then you have this region here, which I'm going to talk about, which is the parabrachial nucleus, which is very interesting. Is going to be a liaison between the input here and the output here in the hypothalamus, as you will see. Other interesting regions are, for example, the central amygdala, which is where contexts are associated with danger. It's because of the CEA, central amygdala, that you feel a night in the forest or a steep slope. Other regions which are involved in anxiety or fear emotions, the BST and the insular cortex. So one thing Ferdinand showed with that experiment is that the PB is really the liaison between the input and the output. And to do that, he injected a AAV expressing CRE in the input in the brain that is in this region called the vagus complex. So this virus is going to follow the information flow from neurons into whatever region, and he injected another virus in the output, but this is a retrovirus. So it's not a retrovirus; it's a retro AAV virus, which is going to go counter the information flow of neurons, and hopefully. And this virus has a fluxed reporter, tomato. Hopefully, these two viruses are going to meet somewhere in the brain, and if they do, they're going to induce expression of tomato in the neuron. And so that was so cool to see that there was one region which lighted up, which was the PB. You can see these neurons beautifully red. The PB was the direct liaison between the input and the output. So many experiments, I'm not going to show you obviously everything. I'm just going to show you one or two of these experiments, the key ones. And this one is the key experiment to show that a particular population of neurons can induce a phenotype you're measuring. So what we did here was to inject this virus expressing a DREAD, and DREADs are receptors which are going to be activated by a chemical, CNO, and you have two forms of DREAD, activating DREADs or inhibiting DREADs. So this is an activating one, and this virus also expresses mCherry to report the cells which express this DREAD. So this DREAD is actually a conditional one. It only is going to induce expression of this receptor in neurons which have been activated by, in that context, by LPS or L1 beta. So once the mouse is exposed to L1 beta, it's going to induce expression of mCherry and this DREAD in the neurons of the region you targeted. So we target different regions, and so the PB, obviously, and the central amygdala for testing, also for control. And then you let the mouse live his happy life. You wait a couple of weeks, and you inject the CNO to assess whether just activation of these neurons is sufficient to induce the phenotype you tested at the very beginning. So that worked. When Ferdinand reactivated these neurons in the parabrachial nucleus, which were activated in the first place by L1 beta and LPS, he induced very robustly this cortisone response via the HPA axis. As a control, when he targeted the central amygdala, this did not work. And we knew already that the amygdala was not a liaison between input and output, but that does not mean that it's not able by itself to induce the response. But here we show it does not. So then once you show that these neurons can induce activation of the HPA axis, he did then for immunologists, kind of a trivial experiments to show that activation of these neurons will induce regulation of the immune system. So this is the thymus, it's the same in the bone marrow of B cells, and activation of these neurons induces a reduction in the proliferation of T cell precursors in the thymus at the DN3, DN4 level. This is of course obvious because we know what cortisone does, but imagine that you would not know what these neurons do. And so in that context, pretty cool to imagine that just activation of these neurons somewhere in the brain does induce an immunoregulation in the periphery, not knowing that the HPA axis would be involved. Of course, then you would look for the mechanism and find out, it's cortisone mediated. But basically it shows that these neurons, just activation of these neurons is sufficient to induce this immunoregulation. So why is it actually interesting or exciting to imagine that? And again, this is hypothesis, we haven't gone there yet. But so the parabrachial nucleus is an interesting region because it's also a hub for different things, which are basically associated with negative input and feelings. So the vagus nerve, which senses malaise, don't feel well, is going to send information to the PB. It is also true for pain. So pain is going to be transiting through the spinal cord, which will also project into the PB and feed the PB with that information. It also goes with memories. Remember, central amygdala is associated with contextual fear, and also this contextual fear is going to feed information into the PB. So basically, the PB is a hub for negative input. If you think that IL-1 beta is a negative input because of what it presents in the immune system, and it's exciting to imagine that maybe the PB can associate, a negative context, like a night in the forest, with an inflammation, and then the PB might make you fearful of a night in the forest because for you it's associated with elevated IL-1 beta in the blood. So then the brain's function is to avoid this situation because it induces inflammation. So again, it's kind of a Pavlovian paradigm which we are testing. So a last question I'm asking here is a lot of colleagues and reviewers that ask this question is, so how is it sensed? So the trivial reply to this question is the vagus nerve, because the vagus nerve is going to innervate all your internal organs. So it's kind of obvious to think that the vagus nerve does sense IL-1 beta and then convey this information to the PB. So we did cut vagus nerve. We did block it at several levels, but there was no change in the phenotype. We also tested the pain inputs. That was not the way the IL-1 beta was fed into the PB. But what we saw using NF-kappa-B GFP mouse was that. So let me just see what I have in the next slide. Yeah, so the AP is the area post-trema, which is a region I mentioned before. It's just under the cerebellum. There's a little ventricle there. And it's a region which is highly vascularized. So this triangle is full of vessels, of stroma cells, and not so neurons, but neuron terminals. When we injected, LPS or IL-1 beta, within 1 hour, we had quite a robust activation of GFP, which here reports activity of NF-kappa-B. So this is an NF-kappa-B reporter mouse. That is, when NF-kappa-B is activated in a cell, this is going to induce transcription and transcription of GFP and GFP expression in mouse in the cell. So within less than 1 hour, the two regions we saw activated in terms of NF-kappa-B was this area post-trema under the cerebellum, and here the median eminence, which is the region just below the hypothalamus, which is full of vessels as well, which is actually the portal system to the pituitary, which would be here. And that's where the neurons, which express CRH, which is the first step in the HPA axis, would actually be somewhere here. The dendrites feeds into that region, and that's where they read CRH into the system. And so what we saw is that here stroma cells do respond very fast to IL-1 beta, and probably release, we're not sure what they release that is going to activate neurons, but we see then that these signals are going to be perceived by the neurons, probably involving prostaglandins, because the whole thing is blocked by blocking COX enzymes, and this then feeds the system. Okay, let me just finish on, it's not directly related to what I told you, but it's just a nice picture. Some years ago, we did a study on depression and microbiota, and what we showed is that the microbiota is determining the amount of arachidonic acid from the food which goes into the bloodstream, And you need arachidonic acid to do a lot of things like prostaglandins, but also endocannabinoids. And you need endocannabinoids to sustain the production of stem cells. And in mice which are depressed, for some reason, the microbiota is going to change, probably, you know, as a consequence of corticosterone increase, because of immunodepression in the gut, and there are going to be inevitably changes. We didn't show that. But so in this depressed mouse, less endocannabinoids, and that led us to look at these the neural stem cells. And it's not really a, you know, it's not a an idea which grew again from the shower or from the bed. It's really because the neuroscientists I'm working with are specialists of these neural stem cells. And the reason is that they're also specialists of the olfactory bulb. And you may know that neurons in the olfactory bulb, because they're directly exposed environments, are damaged easily, and they have to be replaced constantly by new cells, which come from this region and another one. And this is a region of the hippocampus called the dendritic gyrus. What you see are these beautiful cells, are these neural stem cells, and they just, you know, when you, I don't know, each time I look at this picture, it's like, wow, that's pretty cool. I have no idea. I have to ask them what these processes are for. Are they sensing the environment and is a signal to grow or not? I don't know. And so in depressed mouse or mice which have been transplanted with a depressed microbiota, which does not allow the passage for arachidonic acid, you can see there's substantially less of these stem cells compared to normal mouse. It's just, you know, beautiful, and, you know, that feeds my curiosity and my awe for neuroscience. So well, I will stop there. This is my part of my current lab in a lab retreat just 2 weeks ago, and the people who did the specific work I showed you today. So Grégoire Chevalier initiated this study on the Muropeptides and sleep years ago, probably 10 years ago. And this was really taken now to the next level by Ilana Gabani, who has now her own lab in Lisbon, in Portugal. This story about LPS and 1 beta inducing the Christmas tree in the brain was done by Ferdinand Jagot, who then, after his PhD, went to it for 6 months, cycling from Ecuador to the Fuoco in South America. And I didn't put a name here, but I should have. Ziad Al-Nabani here, who did all the work on the winning reaction, and is now has his group at in Bern. Our microbiologist colleagues, Ivo Bonecca here, and Richard Wheeler, who did all the work with us, and our neuroscience colleagues here, Pierre-Marie Dedo and Gabriel Pousset, who is basically coordinating the neuroscience together. And that is it. Thank you very much for listening.
Thank you very much, Gerard. Wow, fantastic work, and thank you very much for sharing this so many great work. And I think so many people listening to this seminar was impressed how much energy you have to jump into a new field and combine your background and like establish a brand new field. So it's time to close, and we want to make a question, but unfortunately in this seminar series, we cannot get the question directly. So if people listening to this seminar want to make a question, please do it through the Twitters. So first search account for Global IMMUNOTALKs and find tweets that say ask question for Dr. Gerard Eberl here, and don't forget to reply to please reply to the tweet with your question, and please mention Global Immune. So thank you very much again, and I hope to see you someday again. So that's all. So thank you very much. Goodbye.
Thank you, Kazuo. See you soon. Bye-bye.
Bye.